Welcome to this lesson. We're going to take a look at transport in plants. So basically, what we're going to study is how various substances move or are transported from one part of a plant to another. For example, water, once it's absorbed here, how does it reach here or how does it reach here? So that's what we're going to study. So this topic is going to be delivered in two sessions. So this is part one. There's going to be part two. Let's get to it. So plants need a transport system for the following reasons. First, to carry water and mineral salts from the roots to the other parts of the plant, like I said. Secondly, to transport manufactured foods from the leaves to the other parts of the plant. So we know that food is made in the leaves by the process of photosynthesis. So once food is made, it has to be taken to the part where it's supposed to be used up. So that's why plants need a transport system. Third, to transport hormones from sites of synthesis to sites of usage. So plants also have hormones. So once hormones have been made, they're supposed to be taken where they're supposed to be used. So the transport system in plants is called the vascular system. So this is made up of two types of tissues that we already know, that is the phloem and the xylem tissue. But there's also a third tissue which is closely associated with these two, called the cambium. We know the function of the xylem and the phloem tissue. The xylem tissue transports water and mineral salts from the roots to the upper parts of the plant, while the phloem transports manufactured foods from where they are made to where they are needed. What about the cambium? The cambium is not really involved in transportation of anything. It is just responsible for production of new cells. So the function of the cambium is to carry out cell division so as to produce new cells. So why are we talking about the cambium? It's because it is somehow closely associated with the xylem and phloem. We're not going to look at that in details, but you're going to be able to understand the physical layout of the xylem and of the vascular system, because the cambium is also involved. So we're going to take a look at arrangement of vascular tissues in dicots and monocots. What are dicots and what are monocots? So dicots and monocots are just two types of plants. We can use this to differentiate monocots and dicots. So mono means one and cot is short for cotyledon. So monocots have one cotyledon. So if this is a seed, there is a part where a shoot comes from called a cotyledon, and monocots have one cotyledon. Then the leaves of monocots also have got this kind of veins which are parallel. Then dicots have got two cotyledons. Di means two. So a seed of a dicot has got two cotyledons in it. Then the veins for a leaf of a cotyledon is like this. They are formed like a network-like kind of arrangement. So this is the difference between monocots and dicots. There are other differences, but our main interest here is not to study monocots and dicots, but to study the vascular tissue. But why are we talking about monocots and dicots? Because the arrangement of vascular tissues in monocots and dicots are different. So we're going to take a look at the arrangement of vascular tissues in monocots and dicots. We're going to concentrate on roots and stems. So start with roots. So we're going to have a cross section of a dicot root. So when you get a root, then you cut it. Then what you are going to see is the cross section of that root. So if it's a dicot root, you should expect to see something like this. So these four here, 1, 2, 3, 4, are root hairs. Then outside the root hair is the epidermis. Epidermis is made of two words, epi and dermis. Epi means upper and dermis means skin. So epidermis means upper skin. Then inside the epidermis is the cortex, and inside the cortex is this here called the endodermis. Endo means inside and dermis, like I said, it's skin. So endodermis is inner skin. So inside the endodermis we have the xylem at the center, and between these, what we can call as arms of the xylem, phloem tissues. So this is how the vascular tissues are arranged in dicot roots. So in short, we can say xylem is located in the center and is star-shaped, while phloem is located between the arms of the xylem. Then for monocot, the arrangement is the same, but inside the xylem doesn't form a continuous star. It is broken on the middle. So that's the difference between monocot and dicot roots. Then for stems, monocot stem versus dicot stem. So we're going to use a table to differentiate the two. So monocots are on this side and dicots are on this side. So monocots are like this and dicots are like this. So if you see, everything is the same apart from of course the arrangement of vascular tissues. In monocots, we have the phloem on the outside and the xylem in the inside. In between is the cambium. So the cambium forms a ring like this, and the vascular bundle arranged in form of a ring, with the inside being the xylem and the outside being the phloem. So vascular bundles are arranged in form of a ring, and well, in dicots, they are not arranged in form of a ring, they are scattered. So if you take a look at each vascular bundle, a vascular bundle is made of 3 types of tissues, that is the phloem, the xylem, and the cambium. So if we take a look at this, we have got the phloem here, the xylem here, and in between is the cambium. So that's the difference between monocot and dicot stems. In monocots, the vascular bundle are arranged in form of a ring. Then in dicot, the vascular bundle are scattered. So that's the difference. So the objective, or what is expected of you, is to be able to label all these parts. So when you're given a monocot, you should be able to tell this is the phloem, this is the cambium, this is the xylem, and the functions of the phloem, the cambium, and the xylem. And also you should be able to differentiate between a monocot and a dicot stem, also a monocot and a dicot root. So now that we have studied the physical structure of the vascular tissues, we're going to take a look at how water and other substances such as mineral salts actually move in the vascular system. So we'll start with uptake or absorption of water and mineral salts. We know that water and mineral salts are absorbed from the soil by root hairs. So water and mineral salts are absorbed by root hairs, which are found near the tips of roots. A root hair cell absorbs water by osmosis and mineral salts by active transport. So water is absorbed by osmosis, while mineral salts are absorbed by active transport. So 2 processes, each is involved in absorption of one particular substance: osmosis, water, and active transport, mineral salts. So I'm not going to go into the details of the root hair cells, because we discussed them when we were talking about adaptation of cells to their functions. So movement of water from root hairs to the xylem. Now that we have studied how water and mineral salts are taken up by the root hairs, the next question is, how does water move from the root hairs to the xylem? The xylem, we know, is responsible for transportation of water from the roots to the other parts of the plant. So how does water move from the root hairs to the xylem? So it moves through 2 processes, one being osmosis, the other one being transpiration pull, which is defined as the pull of water up the plant caused by transpiration. So there's a process called transpiration, which we are going to study, and this transpiration causes the water to be pulled up the plant, and therefore that's how water moves up that's how water moves from the root hairs to the xylem. So here we have studied how water moves into the root hairs, then we have studied how water moves from the root hairs to the xylem. So we're going to start how water moves from the lower part of the xylem to the upper part of the xylem, so that water can be taken to the other parts of the plant. So movement of water up the plant. Water moves up the plant through xylem vessels in a continuous stream known as the transpiration stream. So the way water moves, it moves like a river, like a stream. So it's continuous and it is connected. That's how it moves up the xylem vessels. The forces responsible for movement of water in the transpiration stream are transpiration, capillarity, root pressure, and guttation. So these are the 4 forces or processes that helps water to move up the xylem vessel: transpiration, capillarity, root pressure, and guttation. So we're going to take a look at each of them in details next. We'll start with transpiration, which is defined as the diffusion of water vapor from plant leaves to the atmosphere through stomata. So transpiration can be likened to sweating in animals. So water in the leaves diffuses out of the leaves through the process of transpiration. So how does this help in the movement of water up the xylem vessel? The diffusion of water outside, the diffusion of the water from the leaves to the outside creates a suction force that pulls water up the xylem vessels. How? So let's study this to illustrate. So let's say we have got some water here inside the leaves. So because of transpiration, the water is going to evaporate and diffuse out of the leaves. So here, there's going to be less water as compared to the next part. So this next part maybe is going to be highly concentrated in water as this. Then this water is going to move by osmosis to replace the water that has absorbed, that has evaporated. So here we're going to have less water as compared to this part. So this part is also going to have its water move into this part where there's less water. So this is going to continue up to here. So if there's some water here, it's going to be moved up in an effort to replace the water that has been used up. So this is going to reach the part where there is less water. And if the process of transpiration is continuing, this also is going to be evaporated out. Therefore, this is how transpiration helps in the movement of water up the xylem vessels. So because of transpiration, there is a force that tries to suck water from the bottom to the top. The next process is capillarity. So capillarity is the movement of water into narrow tubes or openings as a result of cohesion and adhesion forces. So water naturally has two types of forces called cohesion and adhesion that makes it able to move through narrow tubes or openings. So cohesion are attractive forces between molecules of the same kind. The word co is usually used to mean working together or things of the same kind being together, being united, like in cooperation, cooperatives and the likes. So cohesion is a situation where molecules that makes up water are attracted to each other, therefore they're always together. Then the opposite of cohesion is adhesion or adhesive force. So adhesion is the attractive forces between molecules of different substances. So water is able to be attracted to other substances other than itself. This makes it possible for water to move up the xylem. Why? Because first it is attracted or it is attached to itself, and it is also attached or attracted to the holes of the xylem. Therefore, it is able to move without falling in the xylem vessel. The next is root pressure, which is the pressure created in xylem vessels due to osmotic gain of water by the roots. So the roots gain water by osmosis, and because of this, there is going to be pressure in the xylem vessel to move water up the plant. So what does this really mean? So if we have a plant like this, and if the upper part of the plant doesn't have water, and the roots have water, so if the roots gain water by osmosis, according to definition of osmosis, it means the roots are going to have a higher water potential than the upper part of the plant. So water will be trying to move to the upper part of the plant, therefore pushing whatever is in the upper part of the plant upwards. So this is root pressure. Finally, guttation is the loss of water drops from the tips and margins of leaves through openings called hydrathodes. So this creates a suction force that pulls water up the xylem vessels. So what is it that I'm trying to explain here? So this is simple, just the same as transpiration. The difference is transpiration, it's because water is going to evaporate outside the leaves, but guttation on the tips of leaves and on the margins, especially in the morning at times, you're going to find some water like this. So this water can drop due to gravity. Because of that, here there's going to be less water, and here if there's more water, it's going to move to replace this water. So this is going to continue, and the water is going to be pulled up, just like the way I explained in transpiration. So we've studied the processes or the forces that makes water move up the plant. So in transpiration, there are three stages that water moves through. So the first is xylem. So in the xylem, when water leaves the xylem, it goes to the mesophil. That's when it goes to air, to the outside environment. So water doesn't move from the xylem to the air, it has to pass through the mesophil. The mesophil is where the stomata are, and that's where water is going to diffuse out of the leaf to the air. So from the xylem to the mesophyll, water moves through the process of osmosis. From the mesophyll to the air, water evaporates out of the stomata to the air. So that's how water moves from the mesophyll to the air. So when water is in the air, it diffuses out to the upper atmosphere. So there are three stages and three processes that are involved for water to move from xylem to the outside environment. So this is what happens to a plant as a result of excessive transpiration. So if the plant keeps on undergoing a process of transpiration, and if the rate of transpiration is higher than the rate of water uptake, the plant is going to undergo wilting. So there are two types of wilting. One is called permanent wilting, and the other one is called temporary wilting. In permanent wilting, when a plant has wilted, no matter how much water you add to it, it will not go back to its normal shape. Well, in temporary wilting, it will go back to its normal shape. So this is what happens when there's excess transpiration. So what are some of the factors that affect the rate of transpiration? So the first one is temperature. The higher the temperature, the higher the transpiration rate. So this can be likened to sweating. When it's hot, you sweat more. You sweat more than when it's not. So the higher the temperature, the higher the rate of transpiration. Secondary, humidity. So this is the amount of water vapor in the atmosphere. So if the humidity is high, it means there's more water in the atmosphere, and this is going to upset the concentration gradient. What this means is the amount of water in the leaves is going to be less, or might be less, as compared to the amount of water in the atmosphere. This will affect diffusion, because diffusion involves movement from high concentration to lower concentration. So if in the leaves we don't have much water as compared to the water in the atmosphere, then it will be difficult for water to diffuse out of the leaves. So we can say the higher the humidity, the lower the transpiration rate. Third, light intensity. So light intensity is just the brightness of light. So they say that when the light intensity is high, stomata tend to open. So we can, because stomatas have opened, therefore more transpiration can occur. So the higher the light intensity, the higher the transpiration rate. Finally, wind. The higher the wind speed, the greater the rate of transpiration. What does this mean? So usually there is some water on the surface of leaves. So if there is no wind, it will be difficult for the water inside the leaves to diffuse out, because there is water already on the surface. But if there is wind, this wind is going to blow off the water on the surface. Therefore, the water inside the leaves is going to diffuse outside the leaves easily. So the higher the wind speed, the greater the rate of transpiration. Finally, we're going to take a look at adaptation of plants to reducing rate of transpiration. Since transpiration can be dangerous at times when there's not enough water, it can lead to wilting. So how are some plants adapted to reducing rate of transpiration? First, they have a presence of a thick wax cuticle. So if we look at this plant, these leaves, so they're shine. This shine comes because there is a wax cuticle on top, which prevents water from being lost. So this is one way in which plants are adapted to reducing rate of transpiration. Two, they have got a sunken stomata. So some plants, some leaves have a stomata which sank down on the leaves. In short, it is hidden as compared to the other parts of the plant. Therefore, it is not exposed to light intensity, sunlight. Therefore, it is unlikely to undergo the process to undergo the process of transpiration, or to lose water through the process of transpiration. Third, reduced size of leaves. So some plants have needle-shaped leaves. Why so? This is to reduce the surface area of the leaf, so that this amount of sunlight hits it. Therefore, not much of it is going to undergo the process of transpiration. Some leaves which are located in places where there is enough water, they have got broad leaves. So those broad leaves, it means the surface area is well exposed to sunlight, and therefore can do transpiration. But for those leaves which are in areas where there's not enough water, they have got needle-shaped leaves. 4. Presence of hairs on the lower side of the leaf. So some plant leaves like chihuahua, or is it pumpkin leaves, they are like this. So these help to trap wind. For example, if it's a windy day, so these are going to trap wind, and therefore like I said, they are the wind speed, they are the rate of transpiration. So if they're trapping winds, it means this transpiration is going to happen. Finally, leaves can roll up when water is scarce. So some plants have got a tendency of doing this when there's no water. So this is to hide some parts of the leaf so that less transpiration can occur. Finally, let's take a look at measuring rate of transpiration. So we measure the rate of transpiration using an instrument or an apparatus called a potometer. A potometer is arranged like this. It has maybe 3 main parts. The first is the plant here, and the second is this capillary tube where water passes, and the third is the scale. So the scale is where you get some readings. So here we're going to have hair water meniscus. So what's going to happen is as transpiration is occurring, then water is going to be sucked here. As water is being sucked, this is going to be moving here, showing that there is some water that is being sucked here. So with that, there's one assumption that is made. We assume that all the water that this plant is sucking is not used for other things in the plant. It is only used for transpiration. In short, the amount of water that is being taken up by the plant directly goes to transpiration. That's what we assume. So what do we get the readings? What are the important parameters to note in this experiment for us to calculate the rate of transpiration? The first is the cross-section area of this capillary tube. So this usually it has to be given because it has to be something that has already been measured. So they will give you the cross-section area of this. Then the second is when you are done with the experiment, this is going to move from here to somewhere. So once it does that, you have to measure the distance it has moved, the length it has covered. So with these two parameters, you'll be able to tell the volume of water that has been used, or that has been involved in transpiration. Finally, you need a stopwatch. So once you start the experiment, you have to record your time, and when the experiment is done, you have to once you start the experiment, you have to start your timer, and when the experiment is done, you have to stop your timer and you have to record that time. Once you have got these 3 parameters, cross-section area and distance moved by the hair water meniscus and the time, you can measure the rate of transpiration using the formula. Rate of transpiration is equal to distance moved by meniscus times cross-section area of tube divided by time taken. So let's take a look at an example. So Mr. Mbote used the potometer to measure the transpiration rate of a leafy shoot of a plant. The water meniscus moved 30 centimeters in 30 minutes. If the cross-section area of the capillary tube was 0.25 centimeters squared, what was the transpiration rate of the shoot? So here's the solution. So just write our formula. Distance moved by meniscus times cross-section area of the tube divided by time taken. So what is distance moved by meniscus? 30 centimeters. What is cross-section area of tube? 0.25 centimeters squared. What is time taken? 30 minutes. So with this, we can compute. So we just put the values. Distance moved 30, cross-section area 0.25, time taken 30 minutes. So when we solve this, we're going to have 0.25 centimeter cube per minute. So one thing I'll emphasize is because these are numbers, they are easy to punch. 30 times 0.5 divided by 30 is going to be 0.25. The main issue here is the units. So the first part of the unit is the volume. So always you have to use the units that you are working with. So if it's centimeters, it's centimeters. If it's meters, it's meters. But this part is volume, so you should have a power 3 here. Centimeter power 3. Then per minute. If you're working with seconds, write per seconds. If you're working with minutes, it's per minute. But usually it's centimeter cubed per minute. So this marks the end of this part of the lesson. Hope you have enjoyed, and happy solving as usual.